Hi, everyone. My name is Tony St. Ledger, and I'm an assistant professor within the departments of ophthalmology and immunology. And today I'd like to tell you a little bit about my lab as you begin to think about your rotations for graduate school. And I'd like to highlight the importance of vision in this graph here that shows public perception about which is the worst condition to endure. And you can see in 2016, blindness was considered concerning to a similar level of concern with Alzheimer's disease and cancer above infectious disease, heart disease, or any other sort of condition. And we know that the visual system relies on multiple components acting in concert to preserve function. So for the visual system, light needs to pass through the optically clear cornea, get focused by the lens, captured by the retina, and delivered and processed by the brain. The focus of my lab is the anterior portion of the eye, or the cornea. So we study in immunological mechanisms that function during ocular surface disease. And the goal of our lab is to manipulate those immunological mechanisms to try to preserve vision during infection. And so my lab focuses on two main projects. The first is related to the ocular microbiome and how it plays a role in disease. So the backbone of this project was from a paper we published in Immunology in 2017, where we showed that inocular resident bacteria, specifically Corynebacterium mastitis, colonizes the ocular mucosa, and it stimulates a gamma delta T cell response, specifically an IL-17 response. That IL-17 recruits inflammatory cells and induces the production of antimicrobial peptides that prevents the infection of blinding pathogens, Candida albicans and Pseudomonas aeruginosa. So the studies that have gone further after this, we are beginning to look at how we can genetically engineer Corynebacterium mastitis to act as a probiotic. We're also seeing how Corynebacterium mastitis functions during times of immune compromise, during times of dry eye disease, and just how it really relates to all of the ocular surface diseases that we know of. And in parallel, we have a project focused on the corneal nerves. So the ocular surface is the most densely innervated tissue in the body, and you can see that by any time you have an eyelash in your eye or some sort of sand or grit, that you can really feel it, and it hurts quite a bit. So what we found is that during ocular herpes simplex virus infection, that those sensory nerves actually retract, and the eye loses its sensation ability, and we get a severe dry eye phenotype that leads to blindness. And what we found is that the factor, vascular endothelial growth factor, or VEGF, controls this neuroimmune axis. And these data were published in an Immunology article just a few weeks ago. And so now we are pursuing studies looking at how VEGF, how the immune system, and how the neuroimmune axis functions within the ocular microenvironment to either preserve vision or to cause disease. And so I'd like to highlight some of the unique aspects of the St. Ledger Lab, is that we're really uniquely positioned to study broad questions related to the immunology of infectious diseases. So we have expertise in the herpes simplex virus infection, and Pseudomonas aeruginosa infection, and the ocular microbiome and things like that. We're also champion studies in immunology of barrier surfaces. So we know that the conjunctiva, the lacrimal gland, and the tear duct all appear to be similar to conventional mucosal immune tissues like the intestine. And the eye is actually particularly well suited to be used to investigate the immunology of barrier surface because of its continual exposure to the environment. And finally, we are well positioned to study basic immunologic mechanisms associated with the neuroimmune axis. So like I mentioned, we know that immune cells, VEGF, and nerves interact within the cornea to either keep visual acuity high or to impair visual vision. So now we're really looking into the basic immunologic mechanisms at play um, after that study. Similarly, the St. Ledger Lab has ongoing collaborations with experts in virology like Kip Kingington, microbiology with Rob Shanks, bacterial evolution with Daria Van Tyne, as well as several other faculty within the Department of Immunology. So we're really trying to get this broad sense of we want to study immunology, but we also want to study it within the ocular microenvironment to preserve vision. And the Department of Ophthalmology has a list of unique resources like a state-of-the-art flow core, an imaging core, and microbiology core that really helps with our studies. So we do think we are well positioned to study these really diverse aspects of immunology and diverse aspects of ophthalmology. So if that seems interested to you, you can feel free to email me, and my email address is right here. We have funding. We have some funding from a K99R00 grant. We have an R01 that's getting ready to expire, but we also just recently got a great score on a new R01 that's projected to start in April of 2021. So we're really primed and ready to go and ready to, I guess, to train you. So if you're interested, please email me. Thank you.